Thanks for tuning into my video. My name's Nicole Bush, and I'm going to be describing the summer research I did through an NSF research experience for undergraduates internship. This summer, I was at Johns Hopkins University, specifically at the Carnegie Institute of Embryology in the Joe Gall lab. So, I studied circular exonic RNA in Xenopus oocytes. For those of you that don't know, Xenopus are frogs. And just to start off, I wanted to describe what circular exonic RNA is and how it's made. So as most of you probably know, all of our DNA encodes for genes. These genes have exons, which encode for the actual proteins, and introns, which space out the exons. So normally, when a gene is transcribed into RNA, it remains linear, the introns get spliced out, and you get left with this mRNA that just has exons that encodes for a protein. What I studied is a novel occurrence that can happen where, within 2 introns of a gene, you have inverted repeats. When these inverted repeats get transcribed into an RNA, they'll anneal to each other because they're perfectly complementary. When this happens, you create a new site for the spliceosome to splice together 2 exons that it wouldn't normally splice together. So, instead of getting a linear linkage of exons, you end up getting one exon linked back to a previous exon in a process known as backsplicing. So backsplicing produces a circularized piece of RNA that's only made up of exons, and it's known as circRNA. So circRNA has been known for a while, but its function is still relatively unknown. So what I did this summer was I looked at circRNA and how they exist in the Xenopus oocyte. The Xenopus oocyte is just the egg of the frog. So the first thing that I did was I actually created RNA samples. So one of the great things about using the Xenopus oocyte is that the eggs are so large that you can actually manually dissect out the nucleus, and then you can get really, really pure fractions for figuring out what RNA transcripts are in the nucleus or the cytoplasm or both. So I was able to remove oocytes through surgery, and then I manually removed the nuclei and the cytoplasm, and I also took some RNA samples of the whole cell. So after this was done and I had my pure RNA samples, I then created a protocol in order to identify the circularized version of one gene. So we ended up picking the LSM1 gene, and we picked this gene because in our RNA-seq data, we found that it has an abundant circularized RNA. In order to prove that this really was a circRNA, we had to make sure that we could detect it in the cell. So what we did is we created inverted primers that will only show the circularized version of the gene. So normally when you want to get an RNA product of a gene, you'll make your primers face one another. Because when each primer creates its strip of DNA, those 2 strips will anneal to one another and you'll get a product that cannot be degraded. So what we did instead of creating inward-facing primers is we created outward-facing primers. The reason we did this is because when these 2 exons circularize, you'll actually end up getting a sequence of this novel exon-exon junction that would never occur in the linear version of the protein. So we created these inverted primers, and then I ran a gel, and this gel shows that you do indeed have a band of the right size for the product that we were trying to get. So we ended up getting a band of the right size, and then I sent it out for sequencing, the product that we got, and we ended up indeed seeing the sequence of this exon-exon junction that would never occur in the linear version of the protein. So this was sort of nail in the coffin. It's definitely a circRNA, it's definitely in the cell. And then the next thing that I did was I actually used those pure RNA fractions to figure out where in the cell this circRNA was. So this plot shows both the linear version of the LSM1 gene and the circular version of the LSM1 gene. Now normally, the linear LSM1 gene will exist in both the cytoplasm and the nucleus because it's transcribed in the nucleus, exported to the cytoplasm. The sort of really interesting part of our research was we found that not only is the linear version of LSM1 found in the cytoplasm, but the circular version of LSM1 is also found in the cytoplasm. And the reason that this is a really cool thing to discover is that the Xenopus oocyte has never gone through mitosis. So that cell has never divided into 2, and therefore the nuclear membrane that encloses the nucleus has never broken down. And because the nuclear membrane has never broken down, and we see the circ LSM1 in the cytoplasm, that shows us that the Circ-LSM1 got exported out of the nucleus and into the cytoplasm. And while this doesn't necessarily prove function, it does suggest that it has some sort of function, because the organism had to evolve the ability to export that circRNA. So this was the first half of our project. The second half is a vector design for ectopic expression of Circ-LSM1. So all of our research was done in Xenopus tropicalis. What we hope to do is create a vector to ectopically express the Circ-LSM1 in Xenopus laevis, because this species of frog doesn't have this Circ-LSM1 gene. And then by ectopically expressing the Circ-LSM1 gene, we hope to figure out what sort of function it's performing in the Xenopus tropicalis oocyte. So, our discussions and conclusions, just want to reiterate that we discovered that LSM1 circRNA is present in both the nucleus and the cytoplasm of Xenopus tropicalis oocytes, and because the nuclear membrane has never broken down, this shows that the Circ-LSM1 gene is exported into the cytoplasm. Our future questions include understanding how the circRNA is exported into the cytoplasm, and finding ways that circRNAs function in the cell. Finally, I would just like to thank the National Science Foundation for funding this research and the research of thousands of other undergraduates like me. And I'd also like to thank Johns Hopkins for hosting the internship. And finally, I'd like to sincerely thank the Joe Gall lab. I had so much fun, I learned so much, it was really exciting. And I'm actually continuing this work at my home institution of UC Santa Barbara, which is something I'm pretty excited about. So thank you, and thanks for watching.